This is actually, I think, your 15th Biology of Genomes meeting.
Really? Yeah.
And 45 minus 30. You've been here another 30 times, plus doing things at the DNA Learning Centre. So Svante is I hope this Cold Spring Harbor is a special place for him, and we'll come back to that in a bit. But to begin at the beginning, Svante, how did you get interested in science? Did you come from a scientific family?
Well, I did actually. So my mother, I grew up with my mother, and she was a chemist and had a very sort of high regard for science. And I grew up alone with my mother because my father had another family. We were the secret family, so to say. He had an official family, and he was also a scientist and a biochemist, and discovered prostaglandins, which is sort of a signal molecule which is very important in human reproduction, for example. And he won the Nobel Prize in medicine exactly 40 years before me. So it was almost spooky this fall that on the day 40 years earlier he had won it. But I do stress that I think it's my mother who had the biggest influence on me. And I think it's something rather special growing up as a single kid with a single parent, that you're taken much more seriously in that situation. And when I compare to our kids now, I remember my mother discussing trivial things for the family, such should we buy a new laundry machine, where should it be a more expensive one or a cheaper one, and things like that. Things we would never discuss with our kids, right? They would even not notice there was a new laundry machine. So I think you somehow get taken seriously early, and as did my interests in archaeology and things like that.
And so you did a medical degree first?
No, I actually had this idea that I wanted to become an archaeologist, and particularly Egyptology, and started studying that at university. But since it was this sort of interest since I was a little kid, I think I had a far too romantic idea what it would be like. I thought it would be something Indiana Jones-like, where we would excavate and find mummies and pyramids. And at the end, you had to study ancient Egyptian grammar. So I got disappointed, disenchanted, and then didn't quite know what to do. And then I started studying medicine, because I said, you at least get a job. Okay.
And did you enjoy studying medicine?
Yes, more than I almost thought. I think I started medicine also with a view to doing science and research, actually. But once I met patients, I found that it was more rewarding than I had expected. And that is a sort of privilege. You get to meet everyone in society. In almost every other activity you do, you're in some kind of a bubble. Otherwise, I meet mostly other scientists in my life. But as an MD, I get to meet really everybody. And that's a privilege in a way.
But you didn't continue as a physician?
No, I before I would do my internship, and so I said, okay, now I should do a PhD, because I was interested in research, and I could then come back to the clinic. And that's where we are. I never came back so far. Yeah.
And you did your research, PhD research, on a topic that was interesting, great interest to Cold Spring Harbor in the 70s and 80s. You worked on adenovirus. So you're working on adenovirus, which is it's an important virus. But how did you come to start studying ancient DNA?
Well, so at that time, they were cloning DNA in bacteria from different organisms, from viruses or from whatever organisms, was a rather new technology. And one was sort of still amazed you could do this. You could move the DNA into the bacteria, grow up the bacteria and get lots of it and determine DNA sequences. And of course, from my Egyptology studies, I knew that there were hundreds and thousands of mummies of humans and of animals in museums in various places around the world. and also in Uppsala where I studied. So the idea was not so far-fetched, I think, to say, could one extract DNA also from long dead organisms and study that?
But this was almost a black project. You kept this as a sort of sideline hidden from your supervisor?
Yes. I mean, my thesis advisor was a person I was, I'd say, a great respect for, to put it positively. So I was sort of afraid that if he found out about this, that he would say, this is not what we have our grant money for, things like that. So it was something I did, late at night or at weekends when few or no one would notice, I hoped.
And you didn't even put a label on the front of your notebook for this research, just your name?
Yes, it was a separate notebook for this, yes.
But you kept locked in the bottom of the drawer? I thought your first venture into this was interesting. It involved liver.
Involved?
Liver.
Ah, okay, yes. So, I mean, at the time, there was really the feeling that the DNA was a much more sensitive molecule than it actually is. One was very careful when you handled it with gloves. It was the idea that very rapidly after death, it would be degraded. So I wanted to see if there was at all a chance for DNA to survive after death. So I then tried that out by buying a piece of liver in the supermarket close to our laboratory, and then drying it in the oven in the lab to sort of imitate ancient Egyptian mummification in some way, which is to a large extent drying out the tissues. And indeed, it turned out there was still lots of DNA there. It was sort of degraded to short average size, but there were lots of DNA there. And that's sort of what encouraged me to then go to the museum and try to argue with the curator that one should be able to cut a little piece of the mummies.
And you had an early collaborator in Egyptology that was very kind to you and provided you with a sample.
Yes, so the professor of Egyptology that I had studied for was very sort of open to this and helped me to get samples, not only from the collection in Uppsala, but he had good connections also in Germany, in what was then East Berlin. I went to a trip together with him to what was then East Berlin and sampled, I think, 23 different mummies that they have in their collections.
But you had a bit of a shock when you took them back to the lab and tried to extract. Or in fact, I think you said it was a shock even when you were removing the tissue from the mummies, that you realized how different that tissue was from your dried liver sample.
Yes, so sort of there was indeed differences when you then try to extract it. The sort of the old tissues, sort of the extracts was brownish, and there were sort of things there that fluoresced in UV light, for example, spontaneously. And so there were clearly differences there that I didn't understand.
And so those first set of samples were uniformly negative?
Not totally. What I also did was to look in a microscope in the tissues. And in some of these samples, you could see things that look like cell nuclei, where the DNA is stored. And you could also stain them with dyes that specifically stain DNA. So that looked very encouraging. But then when I tried to extract it, it was in most cases, I had no luck whatsoever.
Was that because it was very fragmented or just?
Probably, or bad techniques at the time, or yes. Mm-hmm.
But eventually you were successful. What changed in your procedures or the samples you used?
Well, I think eventually I was too successful, I would almost say. So I tried to extract DNA from these mummies. And from some of them, I got something that looked like DNA. And then I tried to clone it in bacteria, which was then the sort of technology at the time. And I got some, you put them in a plasmid and there's a selection there. So if the bacteria grows, there is a sort of insertion. So I had something I had cloned, and then I wanted to see if this was human DNA. So I took some human repetitive DNA and labeled it radioactive and screened the clones and found a few that had this repeat in them that is typical of human DNA. So I was very excited about that and published that in Nature in a paper where I showed this microscopic results that there were cell nuclei there that could stain on these DNA sequences. To great fanfare. I mean, it was a big news at the time.
It was even the cover.
It was on the cover of Nature, yes. And single author paper in Nature. At that point, I had to tell my supervisor what I was doing. But then he was sort of rather understanding. It was so.
There was little side story to that. Your first publication of that work was not in Nature. It was in an East German journal.
Yes, I mean, already at that time, you know, we are talking a lot these days about that we have to sort of respect countries and communities where we work and give back. And there was a feeling that it was sort of bad if we come from the West, take the samples, publish in the West. So we took care to have the first publication of the same results, essentially, in an East German journal of the Academy of East Germany. So it was published there, and no one read it. It was absolute. And then 1 year later, we published it in Nature, and it was on the front page of the New York Times, which I think illustrates sort of how badly these socialist countries worked. You know, they could at that point have said, this is a great propaganda. You know, now the New York Times had this on the first page. This was actually published in East Germany a year ago. But no, actually, the secret police interviewed everybody in the museum where we had gotten the samples and sort of said to them that they sort of asked what they had done with us, things like that, and then said they should be aware that Uppsala, the university where we came from, is a well-known anti-socialist propaganda center. And from that point, of course, no one dared work with us again. So yes, it really is how badly those countries work, I think.
And the 1984 paper led to your next position at Berkeley.
Yes, but before we come there, I should say that it worked almost too well, because I'd published those DNA sequences, which I thought came from the mummy, right? And then over the next 3, 4, 5 years, it became increasingly clear that those DNA sequences could not possibly come from the mummy. They must come from me, from the museum curator, from some archaeologist, or something like that, because they were far too long. What we then gradually learned was that this ancient DNA is degraded to short little fragments of 20, 30, 40 nucleotides, whereas what I had published there was several hundred nucleotides. So I think, yes, it's a lesson there that you do make mistakes in science. It's particularly good if you can correct it yourself, the mistakes you did. If others do it, it's also good, but less satisfying for you personally.
Let's stay with the contamination for a moment, although it's off the sort of the chronology. You went to extraordinary lengths to try to prevent contamination of ancient samples by modern DNA.
Yeah, so what became clear then is that there's, of course, very little DNA that survives in tissues that are thousands of years old. Often 100,000-fold, 1,000,000-fold less than there is in modern tissues. And that means that tiny amount of contamination can totally overwhelm the results. Say dust particles in a room like this, where many people are, is to a big extent skin fragments, for example. And one such tiny dust particle can then contain much more DNA than what is in your extract you do. So if such a dust particle lands in your tube, you may sequence just yourself or someone else who was in this room earlier. So progressively became more and more paranoid, a sort of a mania of cleanliness, where they cleaned everything with bleach to sort of oxidize the DNA away, have UV lights at night to destroy the DNA, and change clothes, and had that all the students that work there had to go there first in the morning before they entered any other parts of the lab, etc.
And one reason why that even the small amount on the skin flake could be so devastating sort of arises from when you were here for the talk you heard in 1986 at the symposium on the molecular biology of Homo sapiens, where you gave a talk on the work you've been doing, and in the same session there was Carrie Mullis.
Yes. So Carrie had just invented the polymerase chain reaction, for which he got the Nobel Prize in chemistry shortly thereafter, which is then this method where you can amplify from a single copy of DNA, make billions of copies of something targeted that you're interested in. And that immediately, of course, seemed to be the method of choice to apply to these things. So not try to clone everything, all the molecules that are in such an extract, but just go targeted after something you're interested in.
But I'm right in thinking, in fact, I think in your memoir, you say that your attendance at that meeting, hearing Carrie, transformed your work.
Yes, I mean, I came home and realized I want to do PCR, and started to set that up.
So let's turn now to when you started working on Neanderthal samples. You actually you got a sample from the type specimen that was found in the Neander Valley in cave, in the mid-19th century?
In 1856, yes.
So type specimens are the sort of the gold currency of taxonomy. So how did you persuade somebody to give you a chunk of bone out of this extraordinary remains?
That did take some effort and time. Of course, at the time also could not push, and we still can't promise success with any particular specimen. But we've sort of gone step by step, showing, for example, that animal bones from similar settings as where that had been done did yield DNA. Because it's, of course, also much easier to say work with a mammoth bone or a cave bear bone. When you then find something elephant-like from a mammoth, you're pretty sure you have the right thing. When you find something human-like from a Neanderthal, it could still, in principle, be contamination. So eventually, the curators in Bonn sort of allowed us to remove a sample. And it was also important to me to actually have the type specimen, because paleontology is a difficult discipline. There is a lot of discussion at that time about what is a typical Neanderthal, is this a Neanderthal or not, when you find something. But this is the thing that gave its name to Neanderthals. You know, it was found in Neanderthal. So if this is not a Neanderthal, sort of Neanderthals will not exist. So no one would question that this was a Neanderthal DNA. We got something from it.
And by this time, you devised and developed the techniques that you were using to an extent where you could be quite confident that you would be able to, if there was DNA there, that you would be able to extract it?
Yes, although I should say we are much better today than we were back then. And I think we were also lucky in the sense that what we then studied was this mitochondrial DNA, which is maternally inherited and changed particularly rapidly. And we're lucky in the sense that what the DNA sequences we determined were clearly close to humans, much closer to present-day humans and to chimpanzees, for example, but also clearly fell outside the variation of modern humans that one already knew quite well from thousands of individuals where this had been sequenced. So when it was really immediately clear, it's one of the few cases, I think, in my career when I've seen a little piece of DNA and immediately said, wow, this must be right. Because it was clearly not a human that lives today. It was falling outside of that, but was clearly much closer to humans than to any ape.
So was that initial work just with the type specimen, or were you able to get access to other skeletons to confirm your first result?
First work was just with the type specimens. But we were then still so afraid of contamination, or that people wouldn't believe it, that we sent a part of the bone to another laboratory and had them reproduce it. Mark Stoneking's group at Penn State University.
And they got essentially the same results?
Yes, they got a little piece of the same sequence, and it was sort of similar.
By now then, your reputation for doing this was well established. And did people come to you to ask you to investigate other specimens and other bones that have been found elsewhere around the world?
Increasingly, it got easier to get access, but it's still today, it's the situation that some archaeologists will come and say, we found this amazing thing, can you look at it? And others one have to convince.
Is that because they're afraid that their job's going to disappear into molecular genetics labs, that they wouldn't have to bother about strata and things, you just look at some the remains DNA?
I don't know if there are archaeologists in the room or if I can speak freely. Yeah, I mean, I think what I would say, good archaeologists who are just interested in the questions at hand, they will be happy that there is sometimes then a chance to apply molecular genetic techniques to address these questions. Of course, there are some people in the field, I think, who may have built their career on some hypothesis, sort of before these techniques came around, well knowing that they could probably never prove that they are right. On the other hand, no one else could really prove that they are wrong either. So you could have a whole academic career sort of built around this. And then it's scary, of course, if it comes some new technology around that might just show what is right and what is wrong.
I can remember a similar dispute when DNA barcoding came in between the molecular geneticist doing DNA barcoding and the taxonomist who relied simply on morphological characters, the length of a bird's beak and so on. There was again this same tension between them. So a few years later, you had another interesting specimen to look at. I'm jumping ahead a bit.
Yes. So we worked with this Russian archaeologist in Novosibirsk. They're excellent archaeologists, and I excavate particularly at the site in southern Siberia on the border of where China, Mongolia, Kazakhstan, and Russia meet in the Altai Mountains. There's a huge cave site where they found human remains, they had found Neanderthals, and they sort of sent us specimens that they found. And among those specimens was a tiny little bone that was so small, so it was actually lying around for almost a year in the laboratory with us, not paying attention to it. And eventually we extracted DNA from it, expecting it to be a Neanderthal. And we were very surprised when we got the results, and it was clearly not a Neanderthal. It was very distant from Neanderthals. Went back at least 400,000 years or so to a common ancestor shared with Neanderthals. So we realized that we had sort of stumbled on a new form of extinct humans based on only a genome sequence. So not having any idea how they would look in terms of their skeleton or what stone tools they would make or so. And we debated with our colleagues in Russia at the time a lot what we would call these individuals. And we ended up saying we call them the Nissans after the Nissava cave, just as Neanderthals are called Neanderthals after the Neanderthal site where they were found. And it turns out that they were sort of very distant relatives to Neanderthals. And just as Neanderthals have contributed to people who live today, the Nissans have also contributed in Asia, that people whose roots are in Asia carry 0.2, 0.4% of their DNA come from the Nissans. And in Oceania, so Aboriginal Australians, Papuans, for example, carry up to 5%, 5-6% of the Nissan DNA.
And again, there have been other Denisovan remains found that have confirmed this.
Yes, but not so many. There are other specimens from that site in the Nissava cave. There's a total of 8 different specimens. They're all very small things, teeth and things like that. There is a mandible found in Tibet that turns out to be the Nissan. And there's sediment DNA from sediments in that site that also comes from the Nissans. But that's all the evidence there is still. I'm convinced, though, that many human skeletons that are in museums in China, for example, will turn out to be the Nissans when one does extract DNA from.
So what does this say about the population, the number of Denisovans? Can you make any estimates of how large these populations were?
Well, what we can study is the Denisovan DNA in present-day people. And it's quite interesting if you sort of look on the DNA contributed by Neanderthals to present-day people. It's very homogeneous. There's not much variation in it. It sort of seems to come from a rather small population that mixed early on with modern humans when it came out of Africa. If we look in Asia and this Denisovan component, Josh Ake particularly at Princeton has studied this and others. There are clearly distinct Denisovan populations that contribute to present-day people. So if you look, for example, in Papua New Guinea, where there is so much Denisovan contribution, that sort of contribution is quite distant from the genome we have sequenced, and it's very plentiful. If you look in Japan or in China, there is that component, that population, but in addition, another population that's very close to the genome we have sequenced. So there are at least 2 Denisovan populations that were quite distinct, that contributed to present-day people.
So going back to when you published the Neanderthal study, I mean, it must have raised enormous public interest. What sort of reaction did you get from people saying, "Oh my God, I've got Neanderthal"? You know, I don't. Well, I mean, my image of Neanderthals is rather not very pleasant people, you know, with big brows and you know. Did people say, "Oh my God, this can't be true," or you know? Or...
So, I mean, it was mixed response, but overall it was actually very positive. People seem to be rather proud of their Neanderthal ancestry, actually.
And that's, and presumably by the time you did the Denisovans, that was already, that sort of reaction was not amplified, but that was not unexpected, that people would think that this is really quite cool, that I've got ancient DNA in me.
Yes, yeah, I do think it somehow.
So when you look at the genes that we have that were derived from Neanderthal and Denisovans, they've been around a long time. Does this mean that they have particular benefits to us that to have survived, been selected for?
Well, so say Neanderthals and modern humans separated from each other about 500,000 years ago and then evolved separately. So one has accumulated quite a bit of mutations and differences. And then one meets again and mix these 2 gene pools. So you don't sort of introduce into modern humans and variants that often are a bit more different than other variants. And we are only beginning to learn, I think, about what influence that has. There are some examples of Neanderthal variants that have been studied that has sort of impact today. So there is, for example, ion channel, a gene encoding an ion channel, a sodium channel, that's important for inducing this sense of pain when we hurt ourselves. And the Neanderthal version has 3 amino acid differences in the protein, which is a lot. It's very rare to find a Neanderthal protein with so many differences. And we can show when we express this in cells today that it's sort of this ion channel remains open longer for a certain stimulation. It would seem more sensitive, if you like. And indeed, it turns out that 0.3% of people in Britain carries this Neanderthal version and 1 copy on one of their chromosomes. And if you then look at questionnaires where these people have answered questions about how much pain they experience in their life, all kinds of pain, stomachache, headaches, back pain, they do experience more pain. If you relate that to, this increase in pain we report with increasing age, it's as if they were 8 or 9 years old.
You fairly recently published a paper on COVID resistant, a gene that confers some degree of resistance that you think is derived from Neanderthals.
Yes.
Has that stood up?
Yes. I mean, so when the pandemic came around, we were peripherally got involved in a big international consortium to look for factors in people that contribute to the fact that some people become very ill and even die when you're infected by the virus, whereas most people, as we know, have
It is.
Whereas most people, as you know, have no symptoms or very mild symptoms. So one looked sort of in the genetic variation among people who became very ill and even died, and people who had very mild symptoms. And when we looked at the results of that, there was a very clear signal of chromosome 3, a sort of very unusual for being a genetic signal. It was a very strong signal there. And the variant that confers susceptibility to severe disease turned out to come from Neanderthals. So we were extremely surprised about that. I almost fell off my chair when I saw this. So there is this variant that comes from Neanderthals that about doubles your risk to become severely ill or even die if you're infected with the virus. And it's sort of was very shocking to us, actually. So there's a much interest in now studying. If you compare this risk variant with the protective variant, there are 14 differences in this. So it's a big interest in studying the function of those differences, of course, because if we could understand why certain people become so much sicker, we might be able to treat the infection better also. And you can, of course, look at the distribution, how frequent it is. In Europe, about 16% of people are carriers of this risk variant from Neanderthals. In southern India, it's about 50%, and it's absent in China and Japan, for example. So it's a very interesting distribution around the world. We can estimate how many extra deaths we have had in the pandemic due to this Neanderthal contribution, which is at least 1 million, probably much more, actually. One shouldn't joke about this, of course, but in some sense, it's a late revenge of the Neanderthals. They were probably in the order of 200,000 or so when they became extinct. And now we have had more than 1 million deaths due to this contribution.
I guess we should draw to a close, but I will say just a couple of weeks ago, you were talking about the improvements in technique. There was this paper on isolating DNA from a pendant that had been worn by a woman in, I can't remember what period it was.
Yes, so that's around 26,000 years ago. So that's a sort of It's a pendant that someone had been wearing around the neck. And a group in our institute was able to extract both the DNA from the horn, from the deer that it was made from, and from the person that had presumably worn it, and could then reconstruct the mitochondrial genome so well, so they could date by the sort of lack of mutations that you see, both the deer and the carrier. And the two dates sort of agree even about 25, 26,000 years ago. And it's quite interesting, of course, for archaeology that you can begin to say, for example, was it women or men who did certain things, maybe used certain tools or wore certain things. Now, I think one should also say that that pendant was excavated and taken from the excavation with the dirt around it into the laboratory. Say things that have been in museums and handled by people you can unfortunately not use in this way. But I think for the future, it's going to be important in archaeology.
Well, we must draw to a close, but we've mentioned Coldspring Harbor Labs several times. Can I just ask you to make a few comments, particularly for this group, on the role of the labs meetings in your life and in the life of many scientists?
Wow, well, I mean, it's strange in a way that I have never worked at Coldspring Harbor or done experiments here, but it still feels a bit like home, because when I come here so regularly, it is a sort of mecca for this type of work. As I said to someone here, it's sort of the biology of genomes meeting that now starts tonight is the only meeting I go to in the world where I expect to go and be a participant. Everywhere else, I sort of expect to be invited and paid for the trip and things like that. Because it's a very cool meeting. It's a place where more junior people present their unpublished work often. So you can really get a feeling for where the field is going. And of course, excellent courses here. I mean, close to my heart is sort of programming in biology course, for example, that we try to send people regularly from our group to. So yes, I think it's sort of center and reference point for much of this field, actually, where we are.
Well, I think maybe we should draw to a close. Svante, thank you very much. Fascinating.